Okay, so what we're going to talk about now are capillaries. And we've already talked about capillaries quite a bit. So let's review just a little bit about capillaries. Remember, capillaries, when you look at them, they're very, very, very tiny. They're microscopic blood vessels. You can't see them with your naked eye. And remember, they're only one cell layer thick. All we have there is one tunic, and that's the tunica intima. And remember, they're so small that red blood cells have to go through them one at a time. They have to go through in single file. We also talked about the different types of capillaries. And remember the different types of capillaries, there's 3 of them. And remember we said that they're in beds. So there's a capillary bed right there. The 3 types, remember, are continuous, fenestrated, and sinusoids. So if we look at continuous, remember continuous are the most common type. They have these tight junctions. And so they don't have any holes in them. So when you look at them, they look like this. But remember, these still are leaky. They're leaky. They leak. What they leak is the fluid in the blood. So the water of the blood plus the salts and things like that. Anything that can diffuse through these membranes, but it has to be very, very, very small stuff in order to get out. But they leak. They're leaky. And then we have another type. Remember, these are called fenestrated. Fenestrated have holes in them. These holes are called fenestrations. Remember, that word means windows. But a better way to think of it is like a pore. They have pores in them. And so, because they have these holes, these are more leaky. They're more leaky than those other ones. And if you look at them, they look a lot like something you might have in your kitchen. They look like a kitchen strainer or a colander. And just like you use a colander or a strainer to separate things by size, that's exactly what these capillaries do. They're found in places like the kidney. And so bigger things can get out or in because they can go through these holes. They don't have to go through here. But these holes are not big enough to let things like proteins and cells and stuff out. The holes are still fairly small. Look, there's a red blood cell. You get an idea of what the size of these holes are. But they are more leaky than continuous capillaries. And then we have a third type of capillaries, and remember, these are called sinusoids. And sinusoids have huge holes in them. Look how big these holes are. They're very, very large, which means that very large things can get out. And so things like proteins, even some cells. Now we're going to talk about blood cells today. Red blood cells really can't get out. But we also have white blood cells, and white blood cells can actually squeeze through these holes and they can get out. Another thing you find in blood, which we're going to talk about today, are platelets, and platelets can also get out. So much bigger things can get out. So these are the most leaky. So we have leaky, and then we have leakier, and then these are the leakiest. They're the most leaky. So they're capillaries. Remember, capillaries come in beds, and you can have anywhere between 10 to maybe 100 in a capillary bed. I don't know how many's here, but there's certainly more than one. So if you think about this, these are those capillaries, and capillaries are either leaky, more leaky, so leakier, or there are those other ones which are the extremely leaky, the leakiest capillaries. All of them leak fluid. So as they move, the blood moves through here, it pushes against the wall of the capillary, and fluid comes out. Well, what's going to determine how much fluid comes out is what the pressure is pushing against the wall of the capillary. If the pressure against the wall of the capillary is high, we're going to get a lot of leaking. If it's not very high, we're not going to get very much. But they all leak. And what they leak into are your tissue spaces. So if you look at this tissue that's going, these capillaries are going through, that's where the fluid accumulates. Now these are not leaking, obviously, because there's nothing in them. And so remember, what regulates blood flow through the capillaries are these little precapillary sphincters. These precapillary sphincters are closed, and so no blood can get in there. But these are wide open, and so blood can flow through easily. Remember, we don't have enough blood to fill up all of our blood vessels, so we have to pick and choose where the blood is going to go. So sometimes capillaries have blood in them, sometimes they don't. Also remember that either way, blood still has to get from the artery over to the vein. It has to circulate. So if it can't go this way, there has to be another way for it to go. And remember, this is called a vascular shunt. So, any questions about any of that? None of that is new. That should all be a review from when we talked about it before. But let's talk about capillaries. So again, they're leaky. So a good way to think about a capillary is to think of a soaker hose. I don't know if any of you actually know what a soaker hose is, but you can go to Home Depot or someplace like that and you can buy a hose that fits to your faucet. And that hose has millions of little tiny holes in it. It's just full of little tiny holes. And so what happens is the water goes through there, some of that water leaks out, and you use it to water your plants. So it soaks your plants. It's a soaker hose. But think about this. If this is a soaker hose and this end right here is wide open, most of the water is not going to come out those little bitty holes. Most of the water will go through the pipe, ignoring all these little holes, because it's easier to go through the big opening at the other end, and most of the water is just going to zoom out. But think about what would happen if I took and plugged up this hole. So I'm going to plug it up that much. Well, look, our soaker hose, the water's still coming in here from the faucet. The water's still coming in, but now not quite as much can get out. And so the pressure in here is going to increase. And if the pressure in the hose goes up, fluid is going to begin to come out of these little holes. So still, most of the water is going to come out there, but now some of it's going to begin to come out these little holes. Does that make sense to everyone? Well, what if I were to take and plug up this hole even more? So now it looks like that. What's going to happen to the pressure inside this hose? The water's still going to keep coming in. Not as much can get out there. So what's going to happen to the pressure? The pressure is going to build up even more. And so if more pressure, what's going to happen is more of the fluid is going to cut these little holes. More water is going to drain out through this. And if I plug it up completely, so that there's no way for water to get out this way, the pressure in here is going to be very high, and the water is going to be forced out like crazy. So look at this. What determined how much water leaked out of this capillary, or leaked out of this soaker hose, was what the pressure was. Does that make sense? So capillaries are a little bit like that. So if we look at a capillary, so here's an arteriole, there's our capillary, and here's a venule. Looks sort of like this. So there's our arteriole, here's our capillary, and there's the venule. And look, it goes through tissues. So there are cells right here. These are cells. And remember, the cells are no more than 2 or 3 cells away from a capillary. So all your cells are like this. They're close to a capillary. So what's going to happen is fluid is going to come into the capillary. And remember, we have blood pressure. And the blood pressure in the capillary is still fairly high. It's about 30. So there's a lot of pressure. And remember, there's leaky. So when that blood comes through there, now red blood cells are way too big to get out. So the red blood cells stay in and they go this way into the vein. Proteins, white blood cells, most of those just keep on going into the vein, but fluid can leak out. And if the pressure in this capillary is high, like it is 30 millimeters of mercury, fluid is going to leak out. And so fluid is going to leak out of this capillary. Well, look where it's going to wind up. It's going to wind up where the cells are. It's going to wind up in the tissue spaces, and it's going to become what we call tissue fluid. That's the same thing as interstitial fluid. It's this fluid between the cells. But just like our soaker hose, if we change the pressure, we're going to change the amount of fluid that leaks out. If we decrease the pressure, less fluid is going to leak out. If we increase the pressure, more fluid is going to leak out. Does that make sense to everyone? Just like our soaker hose. So when we look at it, here are those capillaries. Here are the cells. The cells are here and here and here. Here's the cells. And so as that fluid goes through there, or as the blood goes through there, fluid is going to leak out. And if it's a fenestrated capillary, more fluid is going to leak out because it's got all these holes, but it's going to leak out. Well, if we want to know how much fluid is going to leak out, we have to figure out the pressure that it's under. But it's not just any pressure, it's what's called the net pressure. The net filtration, that means it's going to make it come out, pressure, or the NFP, the net filtration pressure. Well, if we look at this word, net is when you add up all of the things and you subtract all of the things, and it's the final result. So if we want to figure out net filtration pressure, we have to figure out the 2 pressures first. We have to figure out the pressures pushing the fluid out, but there might also be some pressures that are pushing it back in or preventing it from coming out. And if we want to figure out this net pressure, we have to subtract those 2. So if we want to know the net filtration, remember that means to come out, pressure, we take the pressures out and we subtract the pressures back in. and we're going to get a number. Does that make sense? So we're going to do pressures out minus pressures in. Well, look at this. What if, when we do it, what if this number over here is equal to 0? In other words, the pressures out and the pressures in are exactly the same. So look, here's a membrane. This pushing this way and this pushing this way are exactly the same. What do you think is going to happen? Is any fluid going to come out? No. Is any fluid going to go in? No. Look, it's 0. So if it's 0, that means that what we got here is no filtration. That's not going to happen. No fluid is going to come out. But there's also not going to be any fluid go back in, and that's called absorption. So there's not going to be any filtration. There's not going to be any absorption. The fluid in the capillary is just going to stay exactly where it is. So when the blood goes through there, the pushing out and pushing in are the same. There's not going to be any movement, any net movement. But what if the number was not 0? What if when we check it out, we take pressures out minus pressures in, what if that number is greater than 0? In other words, pressures out is bigger than pressures in. So look, it looks like this. So now what's going to happen? If our net filtration pressure is greater than 0, what's going to happen? Is fluid going to come out or is fluid going to go in? That's right, we're going to get filtration. So if the number is greater than 0, filtration is going to happen. In other words, as that fluid, that blood goes through the capillary, this pressure is bigger than the one coming back in, and so fluid gets forced out. But look, what if that's not the case? What if when we do this equation, the number is less than 0? If it's less than 0, that means the pressures in are bigger than the pressures out. What's going to happen then? Are we going to get filtration or are we going to get absorption? We're going to get absorption. So that looks like this. Look, this is forcing fluid into the capillary. So the pressure this way is bigger. So fluid comes back in. Does that make sense to everyone? It's really important that you understand if the net filtration pressure is 0, we're not going to get either one happening. No filtration is going to happen. And we're also going to get no absorption. In other words, they're pushing with equal pressure. It's not going to go either way. But if the net filtration pressure is greater than 0, filtration pressure is greater than 0, we're going to get filtration. In other words, fluid's going to come out of the capillary. But if the net filtration pressure is less than 0, it's negative. Fluid's going to come back in. We're going to get absorption. Does it make sense? Well, if you have your page, your worksheet, write this down. It's the very last thing on the page when we're talking about net filtration pressure. Write this down, because that's the way we're going to figure out what's going to happen. And this part right here is really important. So, Once we figure that out, once we know this, all we have to do is figure out the pressures out, because remember, we're going to subtract the pressures out, and then we have to figure out what are the pressures back in. Well, there are 3 pressures, and here they are right here. These are the 3 pressures. Pressure out, there's only one of them. It's blood pressure, blood hydrostatic pressure, or you can just call it blood pressure. Remember, that's the blood pressure going through the capillary. Starts out at 30 and it pushes against the walls of the capillary. And so it's a pressure out. But there are 2 pressures back in. These 2. One of these is called the blood colloid osmotic pressure. That's just a long term for saying how much solute there is in the blood. Because remember, we've said this before, solute pulls solvent. And so what it's going to do is it's going to suck water back in this way, because there's solute in there, there's salts in there, and all those little salt particles suck water. Like, remember we said sodium sucks water? Well, there's a lot of sodium in here. Water follows sodium. So that's a pressure in. And then the third pressure is the interstitial fluid hydrostatic pressure. The interstitial fluid hydrostatic pressure, what that is remember, we're going through tissues and there are cells out here and there's fluid in between the cells. So that fluid, which is already out here, you've got to push against it. It pushes back. So what we're going to do is we're going to take pressures out minus pressures in, and we're going to get a number. And again, the pressures out are this one. There's only one. The pressures back in are these 2. Now, you don't have to calculate anything. What you have to remember is if it's equal to 0, greater than 0, or less than 0. So what is it? If it's equal to 0, what's going to happen? Nothing. That's right. There's no filtration and no absorption. Water's not going to move in either direction. If it's greater than 0, what are we going to get? And if it's less than 0, what are we going to get? If it's greater than 0, we're going to get filtration. Water's going to move out of the capillary. If it's less than 0, we're going to get absorption. Water is going to move back into the capillary. Does it make sense to everyone? So look, here is our capillary right here. So look, there's our arteriole. This is the capillary, and there's the venule. And we're going to take that equation. Remember, the equation is net filtration pressure, and you should write this down if you haven't already. Remember, it equals pressures out. The only pressure we had out was blood hydrostatic pressure minus the pressures back in. And there were 2 of those. One was blood colloid osmotic pressure, and the other one was interstitial fluid hydrostatic pressure. Again, I'm not going to ask you to calculate these, but what I want you to figure out is what the number is. So look, here we're going to do it on this end. right here. Here are those. You don't have to calculate them. They did it for you. Here's the number right here. It's 10. 10. So 10, is it greater than 0, equal to 0, or less than 0? Ten is greater than 0. So what does that mean? Is fluid going to come out? Is fluid going to go back in, or nothing going to happen? Exactly. Fluid is going to come out. So because it's 10 right here, fluid is going to get pushed out crazy on this area because it's 10. But look over here, we're going to calculate it again. And if we look on the other end, when we calculate it, we don't get 10. Look what we get. We get -8. Not 10, -8. It's negative, it's less than 0. So what does that mean? What's fluid going to do? Is it going to filter? Or is it going to be absorbed? It's going to be absorbed. So fluid is going to come back in on this end. And so look what happens. Fluid goes out on one end and fluid comes back in on the other end. Well, why? Who cares? Well, look, it's not empty out here. There are cells out here. So these cells are sitting here waiting for nutrients. They're waiting for oxygen. They're here, and there's like this. Well, look, on this end, arterial end, remember this blood is high in oxygen. It's red. It's low in carbon dioxide. It's high in nutrients. And it's low in waste products. That's what comes out. So that fluid, let's use red, comes out like crazy on this end, because what was the net filtration pressure on this end? Do you remember? It was 10. So look, this fluid, which is high in oxygen, low in carbon dioxide, it's going to wash over these cells. And what's going to happen is it's going to deliver oxygen. We're going to deliver nutrients. And what we're going to do is we're going to pick up waste products. We're going to pick up carbon dioxide and other waste products. And this fluid is going to change. It's no longer this nice fluid like this is over here. Now the fluid is low in oxygen. It's high in carbon dioxide. It's low in nutrients. It's high in waste products. And it goes through here and so on. And then we get to the other end of the capillary. But we figured out the net filtration pressure again over here. Remember, what was it? It was -8. So what does that mean? What's fluid going to do? It's going to come back in. And so that's what happens. So look, the fluid, which is no longer this nice clean fluid, now it's full of waste products. It's going to come back in to the vein so it can be sent to the lungs and drop off the carbon dioxide and oxygen. And then it's going to turn around and it's going to come back this way again. And when it comes back the next time, it's still going to be now this. High in oxygen, low in carbon dioxide. That's the fluid that's going to come out. And so it's like a tiny little miniature circulation. It's like a little miniature circulatory system. because what it's doing is it's dropping off nutrients and picking up waste products. Does this make sense to everyone? Here's another view of the same thing. So look, there's those tissue cells out there. So on this end, that's the arterial end, fluid is going to come out. And that fluid is going to be nice, clean fluid with a lot of oxygen, a lot of nutrients. These tissues, these cells are going to grab it. And the tissues, the cells are going to dump out their carbon dioxide and their waste products. And so the fluid's going to change. And so on this end, remember, it's not going to come out again. It's -8. It's going to go back in. And so again, it's this little tiny circulatory system. And so that's the way that we're going to deliver nutrients to the cells. That's the way we're going to deliver oxygen. It's also the way we're going to pick up and remove the waste products. So we're going to remove carbon dioxide and other wastes. Any questions about it? But we're not done yet. Let's go back to this one. Remember on this end, it's, what was the number? Over here, the net filtration pressure. On this end, it's 10. What was it on this end? It was -8. So look, fluid came out on this end, fluid went back on this end. But look, these two numbers are not equal to each other. If we look at the net filtration pressure across the whole capillary, it's 10 -8. So the net filtration pressure here is 2. So what does that mean? Does it mean more fluid came out than what went back in, or more fluid went in than came out? More fluid is filtered than is reabsorbed. So in other words, every time blood comes through here, some fluid is going to come out that does not go back in. Some of that fluid is going to stay. So look, here are these cells. If I keep adding fluid that doesn't get picked back up, the fluid in here is going to increase. And what's going to happen to those tissues is they're going to begin to swell. And if we don't do something, over time, they're going to swell a lot. So something has to pick up this excess fluid and move it back to the circulatory system. Something has to do that. Well, what does it? There's our net filtration pressure. Again, more fluid leaks than is reabsorbed. That fluid is going to collect in the tissue spaces. And over time, there's going to be a lot of fluid. In about 24 hours, in other words, one day, this is how much fluid is going to leak out of the the capillaries into the tissue spaces. Look, 3.5 liters is going to come out of your blood. Remember, you only have 5 liters of blood. 3.5 liters is going to leak out. If 3.5 leaks out every day, we only have 5 liters. In just 1.5 days, we would have no blood. We'd have no fluid at all. So we have to return it to the circulatory system. Otherwise, blood volume would go down, blood pressure would go down, and our tissues would swell up like crazy. So to prevent that from happening, there's another whole circulatory system whose job it is to pick up this fluid and return it to the circulatory system. And that job, that circulatory system, is the lymphatic system. So the lymphatic system has 2 jobs. One of them is to make sure that that fluid that was leaked is picked up and returned to the circulatory system. But we also want to make sure that fluid is nice and clean, and that's the other job. If we don't return this leaked fluid back to the circulatory system, it's going to stay in the tissues. And the tissues are going to swell up and they're going to fill up with fluid. Does that make sense? Any questions about it?